You're listening to Innovators, the podcast from Harris Search Associates, where we speak with global leaders in education, research, engineering, and the health sciences, and ask them to share lessons learned as they continue to advance the frontiers of innovation and discovery. Today's podcast will be led by Rick Skinner, Senior Consultant.
There is no shortage of effort devoted to solving the mystery of neurodegenerative diseases. Generations of scientists and physicians have devoted entire careers to try and obtain a better understanding of why the brain and the central nervous system of some persons begin to falter and ultimately fail, while other persons live out their lives unafflicted. In the April 4th issue of Nature, a note of optimism was expressed in an article with the encouraging title, Conquering Alzheimer's, a look at the therapies of the future. New and old drugs and combinations thereof, vaccines and gene therapy are being tested, and for the first time in decades, results offer evidence of progress. But this optimism is tempered by the contrast between the incremental progress made in understanding Alzheimer's on the one hand, and what must seem to researchers the vast landscape of what is not known about the disease. So even as we celebrate gains, the task of gaining a full understanding of what causes neurodegenerative diseases remains a formidable one. For some time now, we have devoted a number of innovators to the emergence of data science as a result of vast amounts of digital information becoming available and being harnessed to increasingly powerful computing. Of late, we've heard from some of the leaders in the development of data science about how the field is changing the way scientists go about exploring subjects that bedevil researchers, even or perhaps because of the vast and growing amounts of data available for analysis. On Innovators today, we have with us Christopher Gaiteri, Associate Professor of Psychiatry and Behavioral Sciences at Upstate Medical University of the State University of New York. Dr. Gaiteri joined Upstate as one of the university's Empire Innovation Scholars after serving on the faculty of Rush University as computational neuroscientist. As he explained, and I quote, "I try to address big data for Alzheimer's disease by taking levels of genes and proteins and other molecules of people with and without Alzheimer's, and from that attempt to find what are the key points, the molecules that are really initiating the brain changes that lead to Alzheimer's." Dr. Gaiteri earned the Ph.D. from the University of Pittsburgh and an undergraduate degree from Washington and Lee University. Before joining Rush University, he was research scientist at the Allen Institute for Brain Science and senior scientist for Sage Bionetworks. Welcome to Innovators. When officials at your new university home introduced you, one noted that your approach is, quote, very novel and powerful in wiping the slate clean and letting the biology tell us what networks may be causative versus symptomatic in the initiation and progression of Alzheimer's disease. What were the origins for this new approach, and how does it differ from what has been a more traditional way of framing research about the onset and development of Alzheimer's?
The origins of my approach are ultimately in biotechnology, which has changed tremendously over the last 15 to 20 years as it applies to Alzheimer's disease. I remember 15 years ago in graduate school, I was working with brain samples from 24 individuals, and we were using microarrays, which are basically microscopic arrays of DNA sequences that latch onto their complement pairs. You scan them with lasers, you can measure how many mRNAs are floating around in the brains of these individuals. And 24 samples with this technology measuring most of the genes in the genome, that was a big deal. And then even just a few years later, I was working with better technology from several hundred individuals with Alzheimer's disease. And now through the major NIA consortiums around Alzheimer's disease, there's actually several thousand brain samples with this technology that are available. So that's really been a driving force in shaping my research and the research of many other people, is just a sort of like explosion of not just the number of things that we can measure, but the number of people in which we can measure them. And that really matters for Alzheimer's disease, because we're finding out that each person may have a somewhat unique route into the disease. But as far as wiping the slate clean, as she mentioned, there is kind of a certain satisfaction to when you have this new technology, just saying, okay, let's measure everything we can measure, and then we have disease phenotypes on top of that, amyloid, tau, cognitive decline, and let's just see which molecular systems are the most related to these disease phenotypes, as opposed to just sticking with a particular hypothesis. And I was saying there's kind of a satisfaction to doing that, but on the other hand, when yours is the slate being wiped clean, it can be a little bit dicey. And I've been through that. When the first data set of Alzheimer's disease that I analyzed, we found a really strong microglia signal. And we tested this in cellular models with Harold Newman, who's in Germany, and it turned out really well. We found this molecule that seems to modulate the activity of microglia, which are the immune cells of your brain in Alzheimer's disease. So when I moved to Rush University, I again discovered this same system, the same microglia system in the brain data we had there. And I was like, great, very consistent. But then there was a moment because I couldn't find the same disease association. I wasn't seeing that microglia were associated with Alzheimer's disease in this brain data. And I was like, okay, someone screwed up the data normalization. Let's go back and make sure that this is done properly, right? And I spent 6 months trying to find that same signal and just couldn't. And I ultimately was forced to say, there's something different about these people. And one sort of obvious difference about the people in this cohort, about the people who donated these brains, was they're almost all priests and nuns. And so there may be something sort of different about the way they led their lives, such that they had much lower inflammation in their brains, or at least the inflammation that was in their brains wasn't related to Alzheimer's disease. So when we talk about wiping the slate clean, again, you have to be really humble and realize that, there's a lot of diversity in this disease out there, and you have to kind of be willing to maybe let some things go. And ultimately, actually, we did find that microglia were relevant in those brains, but it was more of a protein signal rather than a gene expression signal.
Gotcha.
But anyway, yeah.
Just a question about that. I mean, it reminds me of something, I guess it was a year or two ago. It was reported about research that had been done on breast cancer, and they had a very large sample. I know there were 7 countries, and I don't remember the sample size, but it was in the thousands and thousands. And what they found was that within that sample of several thousand, the accuracy of the diagnosis, the way in which they'd gone about predicting the outcome of tests from biopsies, they found that it had about a 90, 94, 95% accuracy. The interesting thing about that was that even with such a large sample, the 7 countries turned out to be a little skewed in a couple of ways. So what I hear you saying is that even when you have a large sample size, and even when you have a fairly large selection of variables for which you have data, even then, there are factors that can skew the research regardless of the power of your analytical tools. Am I right there?
Yeah, absolutely. And that's why it's really important to get a diverse sample set. And just having a lot of samples doesn't mean that you actually have a truly diverse sample set. So another limitation of both of those studies, even the larger data set that I described subsequently, was that there's not a lot of African-American participants. And so, so there's a study now led by Lisa Barnes at Rush that's specifically focused on recruiting African-Americans so that, we can, learn more about the disease and make sure that any solutions that we find are applicable for everyone. So, yeah, I mirror you on that point.
It's interesting to think about that, because I think what we're seeing now is this increasing concern that our sampling techniques, or at least in terms of subjects, is really becoming something that we used to take somewhat for granted, and by not acknowledging the diversity of populations that you can get into. So it sounds as if even some fairly basic research methodology questions still remain as paramount as they always were. Let me ask about the statistical techniques, because I can recall in the past techniques like factor analysis and canonical correlation. They were often used for what I call descriptive purposes. That is, the researchers would put together data sets composed of many variable measures of variables and allow the statistical techniques to sort of sort out which of those variables actually covaried. Now, a common description was that this was exploratory to see if and which variables formed patterns that could suggest relationships to be looked at more closely. As you undertake analysis with your approach using big data in the study of Alzheimer's, just how expansive do you anticipate the range to be of the variables that you include? Do you have a particular notion of why the variables you select ought to covary? Or maybe I can put this differently. Do you have what I call a pre-theory that guides that selection?
Well, whenever I read about a new biotechnology that's measuring some big data in cells, I immediately want to apply it to Alzheimer's disease. So I have to say, since that's how I approach it, I would have to say I fall under the more is better theory of big data analysis. But the caveat being more is better if it's integrated with all of the data that you found previously.
Okay.
And so, for instance, there's several new technologies out there that measure the three-dimensional conformation of DNA. When you see DNA depicted, it's always sort of in these spiraling strands that are just sort of floating there. But actually, we know that DNA is shaped into a fractal. It has loops, and those loops are looped. And when you get gene expression, it actually occurs in these sort of three-dimensional spheres within the loop structure of DNA, so that parts of DNA which are very distant from each other in a linear sense, are actually close together in a three-dimensional sense, sort of acting together and creating gene expression. And so this is like an example of a new biotechnology that has rarely, if ever, been applied to Alzheimer's disease. So I would love to measure this technology in Alzheimer's brains and then combine it with all of the other data that we have. And it would be a fairly agnostic approach, basically. I'd be looking at the data-driven systems that we've identified before and trying to relate them to this, and then relating, this three-dimensional chromosome conformation to Alzheimer's disease. So again, my pre-theory is more is better as long as you integrate it and start to sort of form some sort of cohesive theory. Because at the end of the day, we have to do experiments based off of this understanding of the brain, right? We sort of need to narrow it down to a point and say, okay, we're gonna push on the brain with this particular molecule, or maybe two molecules at most, and then we expect this to happen. So we do need cohesion. It's not just measuring a bunch of stuff. It's measuring a bunch of stuff, but then trying to make it cohesive in a way that we can form hypotheses for understanding disease.
But that also leads me to ask another question that. And I'm speaking now as somebody who considers himself a layperson with regards to neurodegenerative diseases. But it seems to me we've always, in the literature and in much of our public policy thinking, we appear to say that the results of these diseases are, at least in part, a function of aging, such that the aging process alone contributes to, and maybe causes, in part, the onset and the progression of the disease. But as you know, within the last decade at least, there are people in the scientific and medical community who maintain that aging should itself be considered a disease that falls within the definitions of what a disease is. So I guess the question what I want to pose is, from your perspective, does the question of age influence your perspective on how you go about framing analyses of data, bearing on the inception and development of say, Alzheimer's? And let me specifically raise one. How important is it to do longitudinal in terms of when you go to analyze data, that you almost have to have longitudinal in order to get at the effect of aging?
You're right that age is the number one risk factor for Alzheimer's disease, but not everyone who's old has Alzheimer's disease, right? So what is the sort of relationship between aging and Alzheimer's disease? I can tell you from the data I've seen and other analysis that I trust that Alzheimer's disease is a very different process from natural aging. we can subtract out the effect of aging in the brain statistically,
Mm-hmm.
and you still see thousands of genes that are changing in Alzheimer's disease above and beyond just those genes that are changing with age. So it really is very different. But there's a lot of variability in that, actually. So we can build models of what it means to age in terms of the molecules that are changing in your brain as you age.
Mm.
And these models are, of course, imperfect. But the thing about the imperfection is that there's people whose brain is older than you would expect based on, how old they are, or
Yeah.
less old from a molecular perspective. And I remember staring at this one, literally a single data point representing someone's brain who at age 65 was 58 in terms of their molecular age. And I was
Wow.
like, I'd really like to be that person. What is driving that, either that resistance to age or that resilience to the effects of aging. And so that is one thing that we try to study now, is like, when we have these models of aging and Alzheimer's disease, the people who are outliers in those models, like what is actually motivating that? And I think that goes to a question that I get a lot, which is when people hear I'm an Alzheimer's researcher, they want to know what can they do, or what can they do for their children to
Right.
become one of these outliers, to become one of these people whose molecules indicate they're younger than their actual chronological age is. And I think there's actually sort of an interesting finding which comes from epidemiological studies, which is that learning another language, if you can become fluent in a second language, that's one of the best things you can do in terms of promoting the development of your brain, so that when you are older, you've essentially got farther to fall, or you've built up more synaptic connections, and it takes longer for you to decline. And there's some caveats to that finding. I think, well, maybe people who speak multiple languages came from potentially wealthier households. But on the other hand, a lot of times people who speak multiple languages speak them because of difficult circumstances. They've been through war and they've had to immigrate. And so I think those two kind of balance each other out. And so, if we could put the next generation of our nation's children in immersive language classes, that's going to buy them likely a couple of good years. When they themselves have grandchildren, and they're aging, they're going to have higher resilience. I think there's some cool things that you can do to become an outlier in these models. Some of them are easier to accomplish when you're a kid. Put your 3-year-old in an immersive language class, or have them go with relatives who speak another language. But there's still some things you can do even as an adult, which are probably boring to hear. But if exercising a couple of times a week, trying to not be obese in midlife in particular, that seems to really matter. And it really, we can see it playing out where basically, like, if you're not eating a bunch of overcooked red meat, you're likely going to have fewer of the angry microglia in your brain that are chewing up brain cells that they shouldn't be chewing up. So I think there is actually, some hope when we talk about this interface between aging and Alzheimer's. It's not inevitable that you're getting Alzheimer's disease in most cases, and there's really a lot of deviation, some of which, you can actually control even later in life.
It's interesting to me because it suggests that maybe the concept of aging, and I don't want to push this too far, but it strikes me that you could say aging may be a substitute for a whole set of processes that we still don't understand very well. That is, that the passage of time in and of itself, and the fact that we know that some people age biologically at a faster rate than others, that maybe aging isn't of itself sort of a surrogate that for the fact that we don't really completely understand those processes. Is that too much of a stretch?
I think in some cases there's not inevitability. I think that's sort of the main thing. It's for most people, it's really Alzheimer's is not an inevitable process. There are, of course, two major types of Alzheimer's disease.
Yes.
And the one that is unfortunately pretty inevitable is familial Alzheimer's disease, which is earlier onset. It's more severe decline, and you sort of start to experience it around age 55. And so that genetic architecture is very different, though. There's just a couple of genes with a pretty specific mutation that are driving that. Although, getting back to one of our earlier points about the importance of diverse populations, they've actually found some people that are specifically resistant to Alzheimer's disease due to just a single mutation actually in another microglia gene. And they found that population in Iceland, which is sort of genetically siloed from the rest of the world. And so it was actually really important to like have far-flung studies and study these different populations.
Now that's
just
interesting.
because sometimes they can hold secrets.
Yeah.
Yeah.
Fascinating. I want to close with a question that it won't be surprising that it's just of special interest to us in our work. I mean, it's mainly about how organizational and administrative leadership influences the development of scientists such as yourself. No one discounts in any way the importance of individual talent and intellect or a person's willingness to pursue a line of inquiry for over long periods of time. So no one wants to discount that. But we think, and maybe that's the best way we could put it, we think the leadership of institutes and laboratories do play a significant role in creating and maintaining an environment in which scientific research can flourish. In your career, and I realize you're still in an early part of your career, but has the leadership of organizations in which you've worked influenced the way you've developed as a scientist? And if so, are there characteristics of persons who held leadership roles that you would single out as particularly important to you and what you've been able to accomplish?
Thinking back on it, there's been great leaders. There's been selectively great leaders, and there's been some terrible ones. Now that I've sort of fallen, I wouldn't say into leadership, but maybe more administrative tasks, I have thought about this a little bit. And at this point, I've met scientists who are so much better at doing the things that I thought I was good at doing than I am, that I think really the best thing I can do is, protect their time. That means all of the financial aspects that go along with science, the various demeaning things that you have to do, the disturbing amount of uncertainty associated with projects where you've already sunk years of effort, all of that stuff I attempt to shield them from because they're so good at doing science. So I realized the best thing that I can do is, get out of their way and prevent anyone else from getting in their way because they're really, supremely talented individuals. So I guess perhaps, that's my lesson from the management I've observed. But just to sort of drive this point home, one of my closest collaborators is Shinya Tasaki, and I met him because about a decade ago, I was working on a contract for Takeda Pharmaceuticals, which is a major pharma company in Japan. And the guy I was working with there, he was sort of a jokester. And he said one day, I'm going to send you the best scientist at Takeda to train. I was like, haha, okay, I'm sure he's smart, but whatever. He was not joking.
What?
This guy is so good. It's sort of literally one in a million. And, I was supposed to train him, and I've learned way more from him than I'm sure he has learned from me. But when you have this kind of value, the best thing you can do is try to protect them from people who would attempt to derail them. Another example is Yanling Wang, who did something really brave, which is she started up a lab at Rush University that's 100% devoted to testing the latest computational predictions of Alzheimer's disease. So not the historical stuff, but just let's use the absolute latest technology, latest mathematical methods. And if we come up with a prediction that no one's thought of before, instead of just sort of, thinking about it for a while, let's go after it right now. And so again, when you're working with people like that, if I can in some minor way, help with their administrative burdens and just, get out of their way, I really feel like that's kind of the management lesson I've learned. I do feel like there's maybe one area in which I could contribute to management, and that's, again, it's been really disturbing to see this up close sometimes, but there's really rampant sexual harassment in science in America, and it's very disturbing. Absolutely disturbing. And when you think about a significant fraction of the workforce that we're expecting to solve Alzheimer's disease to also be dealing with this, it's just terrible. A lot of times you'll see people who are forced to either endure it or give up, their career because, they spent many years in this hyper-specialized area and there's nothing they can do about it. So actually, for a few years, I've been working on a project called Mesh With, which is sort of a technological solution for the Me Too problem. trying to essentially predict and prevent sexual harassment. So we're hoping to, in the next several months, finally get the app out for this. So yeah, I think, again, that's one area in which I think, I could actually make a positive difference. But otherwise, as far as management, administration, and science, I try to get out of the way of the supremely talented.
Well, let me make one quick comment on the sexual harassment issue. This issue has now become, along with the whole question of how to create diversity in the scientific research community, greater diversity, that those two issues now have come to the forefront. And we're finding that in addition to exactly what you described, that is the leaders who get out of the way and try to make it so that research can get done and people are not hampered by too much. But the other ones are trying to protect individuals who are undergoing harassment and then finding a way to encourage more people from diverse backgrounds to come into science. The good news is on the latter is that it seems to be, early science seems to be, there's some good work being done. On the former, I wish I could tell you that we've seen a great improvement. It still seems to be a serious problem, and the really problematic part about it in many ways is that we don't know the dimensions of it. It may be systemic in nature. It may be episodic, but we don't even know, and yet there's too many biographical stories that can be told about women in particular who've simply given up on scientific careers. And so I want to commend you for the leadership you're showing there. I also want to thank you for your time. I want to wish you well in your newest appointment. I know that you've had to battle COVID lately, so you're obviously, I hope you're hale and hearty. We want to thank you very much for being on Innovators.
It's been an honor. Thank you very much.
Thank you for listening to Innovators, a production of Harris Search Associates. We'll have more insightful conversations with global thought leaders to follow.